and my honor and pleasure to introduce a longtime friend and colleague, Dr. Shan Lu, who's a well-known physician scientist and global leader in vaccine science. He's a professor emeritus in the Department of Medicine at the University of Massachusetts Chan Medical School, and also the chief scientific officer of the Worcester HIV Vaccine biotechnology company. I first met Shan probably more than 30 years ago in the early days of DNA vaccines, where he was one of the pioneers in using the nucleic acid technology for HIV vaccines, and in particular, the concept of heterologous prime boost vaccination, and has developed a very interesting and novel polyvalent formulation that involves both DNA and protein-based vaccines given together. And that's about to enter a phase 2 clinical trial in South Africa, and hopefully we'll hear about that today. Dr. Lu has more than 200 scientific publications. He's the editor-in-chief in Emerging Microbes and Infection and a member of the editorial board for a number of other vaccine journals. He's a fellow of the American College of Physicians and a fellow of the International Society for Vaccines. And critically important for the society, he's been a driving force of HIV ISV since its inception, and his leadership has been instrumental in making the society the success that it is today, and for that we're very grateful. So without further ado, I'll pass it over to you, Shan, and we're excited to learn about your HIV work.
Thank you, Jeff. Indeed, we have known each other for many years. Unfortunately, I'm still working on the HIV vaccine. But at the same time, I think HIV vaccine taught us so much. So I'll be happy to share with everyone the current status of field and my own work. And also, as Jeff mentioned, it is a great pleasure and honor to present as this ISV webinar series. As ISV over the last 16, 17 years has grown substantially. We are very happy and thank all the contributor, organizer, volunteer, and many the participants of today's seminar. So my talk may be a little bit out of a box. Everyone heard about the HIV vaccine. You need a broadly neutralizing antibody. Why I'm saying we should work on HIV vaccine, not just neutralizing antibody, but something else. Okay, next slide. As we all know, HIV is still epidemic, or maybe we can say pandemic. Still, millions and millions of people are getting infected every year. You remember when we had a COVID, every country is shocked. But we have an ongoing 40 years epidemic, HIV epidemic. Every year, still 100,000 people dying. Many of them are young children, even adult. Very important labor force in many countries. So this is a very devastating epidemic. Unfortunately, many of us living, if you are not living in those high endemic areas, you may not realize. So this is still happening. Next slide. At the same time, HIV vaccine, everyone know, I'm sure all the participant, the audience know how hard it is to develop a HIV vaccine. Many of us probably at a different time of our own career participate in HIV vaccine work one way or the other. The end result is very depressing. Here is the list of major efficacy trial, not many, about 10 or so, divided into 2 or 3 major scientific attempts, different strategies. Overall, they all failed. I don't need to go through the detail. I highlighted in the middle, 2009. That's 1.5 decade ago. Time passed very quickly. A trial in Thailand actually achieved partial protection. The end of a trial designed period is about 31%. During the first year, maybe 50 to 60% protection. So that really is out of box because most people did not expect that vaccine would work and somehow work. That's the almost common finding in HIV vaccine field. Many study, you have a great science idea, published in many leading journals with preclinical monkey data. Once they went into human trial, they failed. But this RV144, nobody expected that it will have any chance, but it achieved 30 plus percent protection. The platform they use is a viral vector prime and a protein boost. Next slide. Because that was some kind of hope, I would not go through the whole history of HIV vaccine. Maybe last 10 years or so, or since RV144 and many basic research. What do people in the recent past working on HIV vaccine mainly divided into three major approaches. Of course, the broad neutralizing antibody has always been the leading and the largest effort. That's the dream of scientists because the premise is HIV mutated so quickly, so much. So unless you have a broad neutralizing antibody, you have no chance to have an HIV vaccine. So this idea still holds. At the same time, since we have not developed any HIV vaccine, even partially successful one, so we don't know actually whether this mutation will have the impact of any vaccine. But that's the scientific hypothesis. That is, we have to generate broad neutralizing antibody. To support that is the discovery of many monoclonal antibody isolated from HIV infected people. And those antibodies can have a very broad neutralizing activities in in vitro pseudotype virus assay. So that gives us the support that we need to develop a broad neutralizing antibody, just like those monoclonal antibody from HIV infected people. After Thai trial, people realized other form non-neutralizing antibody may be also useful, FC effector function, ADCC, so on and so forth. Also, the T cell vaccine has always been not just so major, but still a very important part of vaccine world, not just HIV. People have thought about test different kinds of T cell vaccine against all kind of infection disease, especially viral infection. So I will divide my talk into this two or three area later, mainly focus on our own progress and data. Next slide. The first one, the main effort, maybe 80% or more effort funding really is dedicated to the generation of a body neutralizing antibody. At this point, we have 6 or 7 epitopes or domain in HIV envelope. As we know for viral infection, especially HIV, the envelope protein is the target of vaccine development, especially for antibody-based vaccine. So there are many, but the next slides, people have worked on different ways. Many of them did not work. So last 10 years or so, the effort in developing a broad neutralizing antibody have been evolved into major 3 categories. The first one is the use of B cell lineage. This is really based on the finding from one case in HIV infection in South Africa. The patient had a first generation antibody, was not neutralizing. Then they go through different period, different antigen stimulate, and then finally generate antibody. Because the sample from this patient, you have the evolution of B cell lineage maturation. So that gives a great example. How do you follow the viral infection lineage? And this time, maybe just use a vaccine. Unfortunately, I list some example here. This approach basically is more complicated than the one HIV infection cases, we can reproduce that. You gave even the trimer, the idea antigen, the patient or volunteers' immune system does not always follow what you want them to do. The next area is called the epitope. Sorry, next slide, Ted. Can you go back? Sorry. Yeah, the next strategy is epitope specific. For example, on the GP41, there are certain domains and also some preclinical work 1 year, all of a sudden, very surprising in animals. The protection target, the fusion domain, works very well. So people say maybe we can develop that. That's epitope, folks. I will mention the result a little bit later. But again, over the last maybe 10 years, really a main focus is the germline target. This is really based on the knowledge of B cell immunology evolution. The idea is maybe a good broad neutralizing antibody may take several stages. So the idea is we will give an immunogen, whether it's natural or artificial or modified, to bind and the precursor B cell, and then stimulate that. And then once the B cell is moving out of the gate toward, example, like a VRC01, generate something in the future, may develop into the idea broad neutralizing antibody. Then scientists will boost with immunogens increasingly complex. That's take many steps, priming, polish, boost, so on and so forth. So I just list some example here. Next slide. Since there are so many papers and data in this area, I would not go over everyone's work. One of the really the most advanced, in my personal opinion, is led by Dr. Roger Sanders. He published a science paper middle last year, so this is probably the most advanced work, including some of the data has not published, but he presented as the seminars or conference. Next slide. So what they did is because all the germline, the strategy, different group may use a slight different immunogen as the priming. So he used the famous BG5405 SOSIP, the trimer, but he modified the 18 site. Here at the bottom, you can see the exact location, makes them more immunogenic and also engaging the precursor. They have a different version. The final one, they have another GT 1.1 with one additional amino acid. From the imaging itself by EM, they can show that they are perfect trimer, everything. Then they immunize the human. Okay, next slide. The most recent work, really this study is very exciting. The first party immunization, they gave this antigen 3 times, months 0, 2, and 6. So finally, the recipient developed, as they desired, the B cell lineage or B cell receptor that is a monoclonal antibody, targeting at the CD4 site. So the CD4 response, the serum response can recognize this, the modified antigen. But on the other hand, no neutralizing antibody to serum. But they were able to isolate through the larger screening of the donor and then identify some of the monoclonal antibody. They not only target the CD4 binding site, which itself is a major progress, they can activate the naive precursor to generate VRC1 class memory B cell in human, and finally induce antibody. That means monoclonal antibody, not from the serum, okay? They can similar to VRC01 class antibody and even neutralize some white virus. So that's the major progress. Then they are giving, they were happy with the first phase. The same volunteer, they select a few, they had this response. I just want to make sure, even the first part, not every volunteer, just some volunteer had the response. Every volunteer, the profile is different. So they give a boost, give the volunteer a resting period of 1.5 to 2 years. Then they give two more immunizations separated by 2 months. They tried different immunogens. To their surprise, they can give the original, the trimer, instead of this step like a prime, polish, and boost. If they just go direct, give the boost, actually generate very interesting data. That is not only they boost further the VRC1 class B cells, like they find in the priming phase, they also generated some other non-VRC1 B cell, but they are still CD4 binding site because the immunogen they use is a broader envelope. They hit other area. So, and this actually the non-VRC class B cell can neutralize better, have a broader antibody than the VRC1. So this is very exciting. They have not published this part. They present this at the conference. So this probably is the most advanced work from published or public available literature. Next slide. The question is from here, how do we reach the end goal to generate a broader neutralizing antibody? So we mentioned there are three general strategy in this direction. The first one, the stabilizer trimer itself, so basically did not introduce a neutralizing antibody. Maybe some can neutralize the autologous virus, that is same virus strain with or without the sugar, but not very powerful. The epitope focus, like GP41, two groups did that. Somehow GP41 is tricky, as the original basic research suggests that maybe there's an autoantibody components. So they both had a safety issue. One's because of the antigen they use, the other one is the adjuvant. So both study terminated ahead of the planned trial. People try to see whether they can use a different adjuvant. So the work may still continue to some project, but some other projects stopped. As I mentioned earlier, the germline targeting immunogen is probably the most promising approach. The magnitude, unfortunately, of the response, either in individual people or the monoclonal antibody, the affinity or the neutralizing ID50 is still low. But the key challenge is these antibodies are not detectable at the serum level. So still a long way from being a real vaccine. As we all know, mRNA technology become very hot, very exciting, and offered a future opportunity because many modified antigen by the antigen design really difficult to express by protein, traditional recombinant protein approach. So the scientists now use the mRNA to bypass the protein step. So hopefully very difficult to express antigen can be expressed in vivo, especially if you need the antigen to be anchored on the membrane. However, one study used mRNA showed a very bad side effect, urticaria. As some the clinical scientists may understand, urticaria are usually in clinical service. It's transient, but in this trial, some of them actually lasting quite many days. So we don't know this is mRNA related or because the antigen is so unique design. Maybe they have auto immunogen component that we have yet to see. So that's a basic general status of the first effort of generating broader neutralizing antibody. Next slide. And we all know NIH and NIH VRC organize a AMP trial, that is give passive antibody VRC1, the famous VRC1, going to human. Each volunteer will receive a very high dose. The half-life actually is pretty good. But on the other hand, when really in an efficacy trial, they found out even the VRC01 at a high concentration could not fully prevent infection. It's only half of the volunteer can be protected, and the virus, when they analyze further, discover that only the virus you can neutralize in vitro in the in vitro neutralization assay. If they can block that in vivo in human, yes, they can work. But on the other hand, the real life situation in South Africa, in many other countries, the virus are keep evolving. And so the one antibody, you think was further neutralizing at one point, maybe no longer so. So actually, there are 50% of volunteers could not receive the protection. Here, the data published in New England Journal of Medicine, you can see that. So next slide. So this really added more challenge from already a difficult effort in generating broader neutralizing antibody. So VRC01, the lesson number one, if we try to use a passive immunization, then everyone agree that one monoclonal antibody, no matter how magic, like a VRC01, is not enough. You need two or maybe more combination or cocktail monoclonal antibody. This will generate a huge burden in terms of cost, and which industrial partner will be interested. It seems this is a major challenge, and we don't know even the cocktail will work or not. The lesson we can learn from AMPTRY to develop active immunization to guide the vaccine development. That means even you can induce VRC01-like antibody by active immunization, it may not be sufficient. That means you have to generate maybe more than one lineage, one epitope, one target. So the first one took us already 10 years. We have not reached that level yet. Then we have to generate several more. Of course, the lesson, the process, the progress we made, probably we can save for future to develop additional epitope target neutralizing antibody. But as we know, HIV, or in terms of the HIV vaccine development, each target, each class of antibody may be different. So the task remains to be humongous. Next slide. So while the neutralizing antibody work still ongoing, there are a few groups to do the T cell response, even though T cell response data has had a major setback, such as Merck's STEP trial. People thought from monkey data, the T cell response was able to protect. When this trial went to human efficacy trial without envelope, just a purely T cell, did not receive protection. In some way, they may also enhance the infection because high CD4 positive T cell, which may help the HIV infection. So this approach, especially the rhesus monkey CMV, later moved to human, even though in the monkey, the protection reached 50% level, but the immune response was not able to be reproduced in human trial. And later, the company licensed this technology and discontinued this line of work. Next slide. So the other effort in a low level, but still receive a lot of people's attention, as we learned from RV144. There's other functional antibody, but they are not neutralizing antibody. Next slide, please. One important information and knowledge learned from RV144 is not just the 31% protection. It's from that study, the scientists at HVTN identified something called the immune correlated protection. For audience today here, we know vaccine in history we usually don't know what is the immune correlated protection, then guide us to develop a vaccine. Vaccine science has been empirical science for many years. Now, with better technology, both immunology and assay development, human evolution, the genomic research, we now many vaccines try to map what is immune correlated protection. But the reality is many vaccines we still don't know. Of course, we know better and better. So that's why when RV144 was able to identify immune correlated protection, that was very exciting because that can guide us to put our effort into the right immune response we should induce. So from this study, it demonstrates that the 31% protection definitely has nothing to do with all the known neutralizing activity or target. Instead, this was found had a correlationship with a highly variable region called the V1, V2 of HIV. This has a correlationship. Later work further indicate the V1, V2 antibody combination of a CD4 T cell may be a high level of a correlationship of a protection. This topic has received quite a few publications. I just select some examples here. Next slide. As you know, follow Thai trial, the global community organized several similar approach, not based on neutralizing, but based on the prime boost, especially viral vector prime protein boost, namely 702 in South Africa, 705, 706 in Africa and South America. The bad news is there's no protection was identified. But on the other hand, the detailed immunogenicity analysis further confirmed there's a tendency that a high V1, V2 antibody can be related to the protection, especially in combination of a CD4 T cell. There are several papers published. I learned that more paper will come out. So in other words, we may still have a chance while waiting for neutralizing antibody being developed. We may need to further study other functional antibodies. Next slide. So this will be a time for me to introduce the work pursued in my lab over the last 3 decades. We started DNA vaccine as a basic research in early 1990s. Actually, Jeff just mentioned as a pioneer himself. There are several groups we started 30, 35 years ago. And then I found when I finished my postdoc with Harriet, I built my own laboratory. We called the Laboratory of Nucleic Acid Vaccines. You can see how brave we were at that time. And but we also convinced the nucleic acid vaccine, both the DNA and RNA, as great future. So I built that laboratory. We really want to dedicate this technology to study novel vaccines. HIV vaccine as the main target. The reason for that is nobody thinks it's feasible. We have a new technology. Nobody believes new technology also. As we know, without COVID, probably mRNA vaccine is just something nobody cares. So we thought we can use HIV vaccine as a model system to test the potential of nucleic acid vaccines. Very luckily, under President Clinton at that time, he set an ambitious goal to generate HIV vaccine within 10 years. Of course, that goal did not materialize. But on the other hand, as a part of that initiative, promote the global vaccine development, such as NIH-VRC was established during that time, and the first HIV program called the HVDDT, that's an NIH program, was very lucky working with the colleagues at the ABL. We were among the first group receive this program, 2000. And then within the few years, we produce DNA and the protein, which I will explain the detail later. It's not easy from academic lab to have a full scale of DNA and protein, and it's a polyvalent. There's many, many cell lines to generate, and the GMP toxicology, go to human study, FDA review. Very luckily, we had the first generation of PDPHV. We call the polyvalent DNA protein HIV vaccine. That's the nickname for our strategy. The immune response actually was very good, even though that was a trial in single site at UMass. So later, based on that data, we received further funding from NIH. One is somewhat basic called the HIF-RAD, the more clinical or product advantage IPCAVD. So I want to take this chance to say I want to thank NIH and US government at that time, the visionary to keep putting money into this seems hopeless program. Both the neutralizing antibody and my program, many others, the whole world, only US government, to be honest, really is the only organization agency keep putting money into this line of work. As we learned from COVID, the investment, the government investment, the public investment is not just in HIV. The lesson, the clinical trial design, the neutralizing antibody assay, those we've initially invested for HIV vaccine really become effective for COVID, for any future emerging infection. So that's why we, being a basic scientist, we always know the money invested in basic science is not going to be waste. It will bring great benefit to the society. So with the support of HIF-RAD and IPCVD, we made the second generation of PDPHV, especially we modified the adjuvant because in our first generation, we use QS21, which is, we don't know at that time, highly toxic. So we modified that. With that, our safety profile is substantially improved and the immunogenicity very good. At the same time, UMass licensed the HIV vaccine concept PDPHV from my lab to a biotech new startup. With the support of private funding and government, we were able to keep making progress. We launched another trial of the HVTN 124, which is a site funded by NIH. Later, the WHV 138 is funded by private investor, and we had two very important papers from the whole program in 2024. And actually, we have another paper, 2025. So I want to share with you next. Next slide, please. So from the beginning, I being a clinician and also doing vaccine science, I think I have no knowledge to know what is B-cell immunology at this time. But from a vaccine lover, to be honest, many people doing vaccine work start as lover of a vaccine. Vaccinology was not existing. It was mainly a work of big pharma vaccine companies. Last 20 years, we have seen many, many basic research laboratory dedicated to different type of vaccine research. So when I started this work 30 years ago, I said, I don't have any vaccine industry experience. I don't know much of B-cell immunology. My PhD was in immunology, but the most on T-cell epitope. I say maybe just use a common sense. I want to study two technical questions. First of all, we know HIV envelope is highly glycosylated. You throw them into mouse, they don't generate an antibody and very poor immune response. You need to give 3 or 4 times. In contrast, you give a flu antigen or hepatitis B antigen into mouse, 1 or 2 immunization, you have very high antibody. I say if we don't have a high immunogenicity, no matter how great your idea is, you're not going to have vaccine. That's the same issue from the current broad neutralizing antibody effort. Sometimes they can generate beautiful VRC-like lineage B cell isolating by monoclonal antibody. But the serum antibody titer, even not the same neutralizing antibody, titer is terrible. So from the very beginning, I said we should focus on this issue. Without titer, without vaccine. I think anyone work in vaccine industry would agree with us. The second one, we do recognize HIV is a mutated virus. But in older day and several hundred years, we have vaccine developed against a highly mutated virus or bacteria. But we don't have monoclonal antibody knowledge. How did the vaccine field survive? One area is called polyvalent. I say we should work, further along that direction. So let me show you the progress in the last several decades in these two areas. The first one, next slide. So let's go back one slide. You went too fast. Yeah. So this is summarize the idea. I found by accident to some way, we did a lot of different DNA work and different pathogen, not just HIV. In my lab, I work on influenza, CMV, many other pathogens. I found out almost no exception. If you use heterologous prime boost, this term used quite often now, but the people have a different definition. I think the mainstream in vaccinology understanding is the heterologous means different vaccine form or platform. The antigen is the same. For example, in our HIV vaccine, we use HIV envelope as a prime, but in DNA format, and then boost with the protein. That will be very different from just giving multiple times of DNA or multiple times of protein. Okay, that's the definition. We found out the heterologous prime boost, different two vaccine modality deliver the same antigen. In almost every cases, they will induce a better immune response, as simple as you just do ELISA measuring the binding antibody. Of course, there are rules. It's a pathogen specific or vaccine specific. For example, the protein first and DNA boost would not work. There's an immunologic mechanism on that. I will tell you later. Next slide. So in HIV, we selected DNA prime and protein boost. We thought this is very simple because we don't have other antigen involved, like a viral vector or live attenuated vaccine or killed viruses. But our work is different from many other prime boost work in the HIV field. Because we are the group of heterologous prime boost, we developed the antigen in DNA form and protein form from the very beginning. So the antigens are matched. Actually, there are many people doing prime boost. The antigens are not matched in HIV vaccine. I will show you. That's not going to work very well. So the prime, why we use DNA? DNA is very pure. You don't have other antigen in it. That's the only coding antigen is your HIV, for example. So, and the in vivo expression of the protein antigen has the best chance of confirmation, the protein folding, glycosylation, so on and so forth. And also this in vivo, the endogenous antigen presenting pathway can induce a better B cell development. And this is really key. I have slides next. So generate good germinal center B cell response. And then also the DNA vaccine, both T cell antigen and B cell antigen. The T cell antigen is not necessary for protection, but it is useful to helping the B cell development. That's the key. So it's a very beautiful combination, simple, and you don't have to worry adjuvant or anything else. So the boost, why we need the protein? Because protein can instantly give microgram range or 100 microgram antigen to stimulate those B cell URA prime. All of a sudden, they can amplify going to plasma cell or memory B cell furthermore effectively. Next slide. So here, just an example, we published that. You can see in the mouse, either bone marrow or spleen, both the secreted cell or the memory population, the DNA protein is much better. This is a B cell ELISpot. You can see you have a humongous number of B cell being developed, antigen specific. I think if you have this, you don't have to worry about vaccine-induced antibody response. On the other hand, DNA or protein alone is very poor. That's why many vaccines need adjuvant. Next slide. So this is not just a mouse phenomenon. We publish also in human, use our approach, the DNA protein. You can see huge amount of antigen-specific memory B cells. Next slide. And then the second question we try to address is a polyvalent versus a monovalent. And this is a classical vaccinology concept. We feel use DNA as a priming is even more useful for polyvalent approach. As I mentioned earlier, the DNA priming can generate a large number of B cell development. So plus the polyvalent mixture, the diversity of B cell, it will be immense. Okay, so I don't have time to go through that. And then the monovalent limited antigen, you can go narrow and narrow. That's why in history we know the antigenic thing. If you give one antigen first, later, you are not going to be able to expand on the similar antigen from the same virus or virus group. Anyway, so next slide. What we did, how to select HIV envelope formulation, we went through a large panel, 60 to 80 envelope. Many postdoc students in my lab, they have to be involved on this massive project. For 10 years, we finished this. We found out not all the HIV envelopes are created equal. Even though structural biology teach us they are same, they are not same. I don't know why. Let's say we use a standard panel of a shorter type of virus, 16. 3 is a laboratory, 13 is a more difficult virus. We find out only about 15% of the envelope from HIV primary envelope, they can neutralize over 50% of this random select virus panel. Another 50% or 18%, they neutralize 20 to 59% of viruses. The majority, over 66%, actually they have the very poor neutralizing antibody. So we go through the traditional vaccine approach, not antigen structure design. So we want to select naturally certain envelope. They are just more immunogenic and more likely generating neutralizing antibody. Then we mix them together to follow the traditional polyvalent approach. Next slide. We are not just, foolishly follow whatever the vaccinologists many centuries ago just random mixed up. We have bioinformatics. Maybe we can call it AI, but 2 decades ago, we did not know there's something called the AI. So on the top here, we have 16 HIV genotypes based on sequence. They are highly mutated, but they have a trend. They later not generated many, many types. They generated about 16 genotypes. And then on the left side, we have the antigen we selected from screening. Let's say A, B, C, D, and AE. They are the many dominant antigen. D is not, but we just include that. You can see with a single primary envelope, you match with against the 16 subtype. The homology is very poor. That's why you cannot generate HIV vaccine by single HIV antigen. The homology analysis, red color means a high homology, blue color means a low percentage matching. But the interesting habit, that is when you have 2 primary envelopes, randomly mix them together, the sequence, the chances of you hitting more virus subtype increase dramatically. It's very easy to understand, actually. Of course, HIV, no matter how crazy they mutate, they cannot mutate to something else, or flu virus or some other virus. They are still HIV virus in order for them to survive. There is a rule our eye cannot see, but bioinformatics or AI can see them. So now if I use randomly any 3 antigen from this 5 antigen I select, you see the frequency or homology can increase further. Finally, to do the box at the bottom, I have 4 antigen, A, B, C, and AE. So they can match over 90 to 97% of the genotype sequence. Basically, use the traditional polyvalent approach. We probably can also match many highly mutated HIV virus. Whether they have the neutralizing antibody, I don't know. But at least from a sequence variation, from the original concern we have, saying HIV mutate, that's why we have a broad neutralizing antibody. I think we have other pathway we can probably to go through that. Next slide. So how do we know whether our hypothesis is correct or the progress we made in immunogenicity and the diversity? It has to go through clinical study. So with the support of NIH and the HVTN, we did a 6-site clinical trial. Next slides. So we used the 5 DNA, 4 envelope, as I select, mentioned to you, A, B, C, and E. We add a gag antigen because we thought it gave some T cell response, mainly CD8 response. In our first generation, we demonstrate the gag does generate CD8 response, but you need some boost probably or higher dose. But at least they prime the precursor. So for the current study, we will not focus on that, rather focus on envelope. So the protein, we have 4 envelope, match the DNA 4 envelope exactly 100%. Next slide. Here is the protein product. Many people in HIV vaccine, including big pharma, they are reluctant to show you the protein gel, SDS page. And you can see how good the quality we manufactured. I think this is also very important. In basic research, you can do any laboratory expression, but when you go to real clinical trial, the quality, especially protein product, is very important. Next slide. The design, we have phase part A, that is to test protein and adjuvant, make sure it's safe. The actual trial, the part B, we have group we call the T2, the blue color, and T3, red color. The T2 is 3 times the DNA prime and the protein boost twice. And the red color is DNA protein giving 5 times, same time. I want to mention that for our DNA immunization, we don't use LNP, we don't use adjuvant, we don't use gene gun, electroporation. It's purely needle injection in saline. Next slide. So this first part is the binding antibody. On the left, you see the antibody against autologous, that is the vaccine antigen. The titer is very high, reach 10 to the 4 to 10 to the 5th. This is almost never heard about in HIV vaccine trial. And also the breadth is also very impressive. On the right side, you see envelope, no matter from clade ABC, so on and so forth, they all can recognize very well. Here, the blue color DNA prime protein boost may be slightly better than the co-delivery, but not statistically different. Next slide. We did antibody ADCC assay done by Guido's group at Duke. You can see both induce a good ADCC activity. Again, here, the DNA prime protein boost, the blue color, is better than co-delivery. Next slide. We also demonstrate the ADCC can target infectious molecule, infected target cells, which was a challenge in HIV field. Most ADC, they use a peptide or protein loaded on target cell, but not the IMC approach. This is more mimicking a live viral infection. Next slide. We also generating neutralizing antibody, mainly against so-called tier B virus. Those are the primary isolate. They are not T cell or laboratory adopt. but their level in our case is varies, but at least we can see we have antibody against very diverse subtypes. Next slide. We also look at the important parameter identified by Thai trial, RV144. That is the V1, V2 antigen of HIV. Here, we measure both IgG and IgG3. As learned from Thai trial, the IgG3 against V1, V2 is more stringent but a better correlated protection. You can see here we have the lower part, the very good IgG3 antibody. And especially the DNA prime protein boost group is better than the co-delivery group. Next slide. We also measure CD4 T cell response. On the left side, that's the DNA prime protein boost, again, better than co-delivery, the CD4 T cell. This CD4 T cell is polyfunctional, as shown by the right side, against diverse cytokine prototypes. Important to mention that here, this assay is not against our vaccine pool of a peptide. This is against the global HIV envelope. So that means our T cell response is also diverse because we use a polyvalent approach. Next slide. Not only from the human immune response, from serum response or T cell, we were able to isolate some monoclonal antibody from volunteer. This is a very rare. The first one is we identified the CD4 binding antibody. This antibody is very hard to generate, even from animals. Almost before we publish, nobody can generate such antibody. And this antibody is also neutralizing, including a few tier 2 viruses. Next slide. This is work in collaboration with Tongqing Zhou at the VRC and the Peter Kwong's group. You can see we neutralize about 10% of this 208 virus panel. The potency is not high as VLC01, but the breadth actually is better than B12. So that was kind of surprising. And the lower part, we highlighted some tier 1B and tier 2 viruses we can neutralize. Next slide. And Peter Kwong and his colleagues made the EM structure, confirmed that this is indeed a CD4 binding monoclonal antibody, even though the binding angle is different from VLC01. So we identify new type of monoclonal antibody. We use GP120, not trimer. We actually can generate monoclonal antibody against the most important CD4 binding site and with neutralizing activity. Next slide. Not only neutralizing antibody, we also isolate a bunch of monoclonal antibody which has ADCC activity. The first, we published a paper just showing we have identified a panel of potent ADCC antibody. This is published Journal of Virology first. Then we noticed some of them is unique and standing out with very strong potency. Next slide. One of them is from this donor, ABL0301. From their serum, we already see the ADCC activity as shown by the arrow there, done by Guido's lab. Next slide. And we look at the breast on the left side, they bind to different GP120. And also they have the ADCC activity against diverse envelopes. And this is, we use a positive control called the A32. This is like a gold standard in HIV field. The antibody was isolated from HIV patient. Here, we were surprised to find that this antibody is as potent as the A32. Next slide. This is the original report of A32, both with the original data and the structural data. Next slide. So we say, okay, what the difference of this antibody? Are they bind to the same area? So we did a competition assay. Actually found that they are able to compete against each other. Then we say, oh, maybe they share some binding area. Then we looked at their sequence. Amazingly, this antibody gene sequence are very similar, suggesting the antibody 78 from HIV vaccine share the same germline as the original gold standard A32. Next slide. Finally, same lab from Paziger's lab at the Army Uniform Service. They did the same EM, like a crystal structure as the original A32, find they bind on the same site, even use the similar area of amino acid. Next slide. So with all this, last year, HUV10 did an analysis. They say, okay, we want to compare, Sean, your data with 13 other HIV vaccine trials. See the key parameter, that is the V1, V2 antibody, how yours compare with others. Next slide. It's quite exciting to see that the DNA prime protein boost, not the co-deliver one, the group had a much better V1, V2 antibody than almost any other HIV vaccine tested by HUV10 trial. It's a 13 trial, but there are 25 different trial groups, arms. So clearly, this was much better. Next slide. And we also looked at another important question in HIV vaccine field, that is longevity, because the previous HIV vaccine or even COVID, most of the modern vaccine use a subunit. They don't really last very long. So you can see here from the 124 trial, we only had 6 months. Later, we did not collect sample. We found that they are basically quite stable. In later, a follow-up phase 1b trial done at the Brigham Women's Hospital, we followed 12 months. The antibody titer are still pretty maintained elevated. Next slide. So from all this, I think we can say now PDP-HV is a highly safety profile in more than one study, human studies. We generate polyfunctional antibody, including binding, anti-VYBC, and ADCC, and some neutralizing. Of course, we don't have tier 2 neutralizing in the serum. That's not our goal here. We have a good CD4 and long persistent of endurance of antibody response. And most exciting is IgG against V1, V2, and CD4 combination. We have a more monoclonal antibody isolated to support our claim. I realize my time is up, but the things in the beginning, there was some introduction. I will take advantage of that to maybe give a few extra slides. Next slide. So the one question in the field, people asking why the follow-up Africa trial 702 did not reproduce RV144. In other words, is RV140 real? So this is from Peter Gilbert's lab published. The V1, V2 antibody in RV144 is high, about 33%, and that may match with their protection level. But in 702, whatever reason, the V1, V2 antibody is very low, very poor. Next slide. And also, I just mentioned earlier, that is in HIV field, the many prime boosts, the homology between prime and boost are very low, 7 to 80%. But in our case, we use 100% match. Next slide. And also the famous MOSAIC trial in the last 2 major efficacy trial, they did not protect. But on the other hand, our concept is different. MOSAIC is not diversity or polyvalent. They just put different areas of a viral sequence from different isolates into together. So this would be a better T-cell vaccine design, not for antibody. You can see the use of one single protein boost bivalent as the prime So they cannot generate really a diversity. On the other hand, our HVTN-124, we use a full prime and full boost, and they are hugely different as reflected by different color, showing different subtypes. Next slide. And we can skip this due to time. Let's move to the next one. So I guess there is a statement circulating in the HIV field. That is the failure protection in HVTN 702, 705, 706 maybe disprove the RV140 result. More important, the immune correlated protection may be not justified. But from our analysis, you can see that's not correct because this trial did not use the best immune design and they did not induce the most critical parameter V1, V2 antibody. Next slide. And that also gives us the thinking. So we can, on one way, work on the broadly neutralizing antibody. We still have some major objective to achieve. We have to move from neutralizing monoclonal antibody to a serum antibody because of vaccine, you have to go to immune response in serum response. We have to move from one epitope or one type of monoclonal antibody generally to diverse group of antibody epitopes. The key is how do we link or predict in vitro assay neutralizing antibody to a human protection. So currently, I think this is great science research. We need to continue that, but we may have a long way to go before a future effective HIV vaccine. At the same time, I and many other colleagues think that we need to try diverse type of immune response, including polyfunctional antibody, broadly cross-reactive antibody. They are not just ADCC, the V1, V2, the tier 1B neutralizing. They actually against very diverse subtype. We don't know in real life situation, any of this can contribute to the protection, especially the combination of all this by one vaccine, you can induce all of that. Whether the low protection efficacy by any of the approach can have a cumulative or effect to achieve a better protection. So I think the real vaccine development is what we propose to do, not waiting for a fancy approach, induce perfect antibody, but at the same time, can we try different approaches as the traditional empirical plus modern science technology to test the vaccine efficacy in well-managed clinical studies. Next slide. So we are going to, our group, going to test the phase 2A trial in South Africa with the greatest support from South Africa community. As some of you know, politically, now South Africa become a no land. You cannot go there, no funding. I think they need our support. We'll be very happy to put our trial to be done there. This is not an efficacy trial, but just further demonstrate, reproduce the immunogenicity in a different human population, different environment, prepare for the later efficacy study. So I will just stop here. Next, thank the colleagues, the partners who contribute to our work. The study, we did a 6 site plus Brigham Women for the phase 1B trial. And next slide, you can imagine how many people contribute over the years, both HVT and 124 team led by Ian Frank and many others, HVTN Central Laboratory, my lab, and some the basic research as we isolate human monoclonal antibody. We'll stop here. Sorry for a little bit longer, or we'll be happy to answer any question you may have.
Well, thank you, Shan, and thank you for sharing your epic journey. It's quite a tour de force against it's a very challenging problem, but the data looks encouraging. So we do have several questions, and perhaps I can read them out to you. What can AI technology add to DNA prime protein boost?
I think with our work, if in an ideal world, we can generate different modified immunogens to do a small group of human study, test antibody response, especially the B cell lineage profile. I think this is very powerful. But again, AI, I strongly believe that AI cannot replace in vivo or human study. Unfortunately, not in the near future. Maybe someday when we learn more, you give one antigen into human, the human response, you can predict that. Then you can use data to generate AI database. Right now, we don't have that. But that's why the B cell study group, they brought the neutralizing antibody. They are very useful for anyone, including us, to develop better vaccines. I think we can use the structural design, AI as a tool, human immunogenicity study combined together. I think we'll have better vaccines, including AI's contribution.
Thank you, Sean. The next question is, are the polyvalent HIV envelopes on the same plasmid backbone, or are you immunizing with multiple separate constructs?
Sure. This is a basic question about not just DNA, but also mRNA vaccine too. We did the early days, no matter what the platform, actually to express multiple antigen from the same vector. It's feasible, technically feasible, but the immunogenicity probably not very ideal. And since the manufacturing process for four or one, actually it's not much difference cost because if the total dose is fixed, you just make several fermentation. One is make a four, one is make one, you mix them together. It's not the idea. I think at this time, I believe we still have to use different vectors. Yeah, just a full plasmid. Yeah.
And the next one is, do you know which cells are transfected with the DNA in presenting envelope?
This is a question for many years. Maybe Jeff, you know better than me. We don't know exactly, but one thing I can say is many cells in our human epidermal cell, muscle cell, probably many cells can do that. That's the amazing thing of a nucleic acid vaccine. Is you don't need a large quantity of the DNA vector or MI, they can transfect quite efficiently, even small scale, right? Jeff, you have any addition on that?
No, I mean, I think the data that we did back with the DNA vaccines, and I think it's true also for mRNA, that you get both cross priming and direct priming. They both
Yes.
contribute. Neither is, both are sufficient, but neither is necessary. So I think it's broader. Then the next question, what is the mechanism of delivery of the naked DNA since it's administered by injection rather than by electroporation?
Well, there's no mechanism. The mechanism is a human evolution. That is, plasmid or nucleic acid can enter cells, just not efficient. So the answer is how I can tell you. It's any other lipid nanoparticle or electroporation or gene gun I used earlier is we use physical force, chemically, physically to help to improve the probability of nucleic acid into cell. But nucleic acid into cell, I think, is a natural ability of human system. It is there endocytosis or other things? I don't know the detail. That's a very different level of a study. I think someday maybe we can do more. Early days we did, but not can answer all the detail. But I want to mention this genetic information we call the transposon, right? That's a phenomenon. That is, they do cross different species or individual. So that's something we did not understand. We just think they cannot. Actually, they can, but not efficient. Yeah.
And the next one, I think you alluded to it. Is it plausible to give the protein vaccine as the prime and the DNA as the boost? In our own hands, we found that that just didn't work as well
Yeah.
as starting with DNA.
Yeah. So this is very easy to explain, as I mentioned in my talk. DNA or mRNA, actually, we published that mechanism, mRNA now publishing more. That is, we induce a B cell. This is very important. Let's say your ultimate goal is a high titer antibody response. You need to have more antigen-specific B cell to be activated. You have a plasma memory cell pool. Then you give a protein, they can, transform further into antibody secreting plasma cell. You give a protein is exogenous antigen. They can stimulate whatever you have there, just like original protein-based vaccine. So you cannot stimulate have more B cell. That's why it's not going to be effective. And then use DNA to boost because DNA is not antigen. They still have to be expressed. The level is a sub-microgram range, but the protein you can deliver in 1 second, microgram, even tens of microgram, 100 microgram into human. So the mechanism is completely different. So you need to have that sequence. Yeah.
Right. Next question, did you look at mucosal antibodies?
Yes, we did. Actually, the DNA prime can prime mucosal system very well. We don't have a detailed analysis, but we did influenza study with MedImmune. We published on that. We did the cholera GI tract study with a group in China. We published that. That's something we plan to do. I know very well. We learned, mucosal immunology many years ago, saying they are all connected. But how do they connect? Actually, we don't know. So from DNA immunization, we find that we can use DNA immunization to connect them. So that's a very fascinating. The systemic and the mucosal actually can be connected. After COVID, there's a tendency in literature, people blame no mucosal immune response or no mucosal delivery is the reason of a failure of many vaccines. I do not agree. I think we did not induce the right type of combination, systemic and mucosal. So mucosal delivery is not absolutely necessary.
Next one is, do you think RNA prime protein boost could have some advantages?
Yes, we have a patent on that.
Have you directly compared the two and shown that one is better than the other?
I think in the same, because RNA we know with LNP, the dose it has advantage. You need less RNA versus DNA. But on the other hand, it's not a fair comparison because DNA I use a saline needle injection. mRNA use LNP, right? Without LNP, it's not stable. You cannot deliver very well. So in that situation, a naked needle injection versus LNP facility mRNA is more effective. But on the other hand, mRNA has some, let's say, low-level tolerability, reactogenicity issue, right? I take a mRNA COVID vaccine, I take a Tylenol every time, ask my family and my patient to do that. You don't have to do with the needle injection of DNA. Also, there's a lingering concern like heart and muscle damage for MI to certain age group and other things. LMP, I'm not worried of mRNA vaccine, but I'm more worried of NLP. Now, everyone is fascinated about that, but my view toward vaccine is a general population, intervention. If I can save have less material involved, I would prefer to do that. So I think for purely technology, I think mRNP prime is very good. But in terms of long term, what the society eventually will accept that, we don't know. So even we talk about the US government, they are misguided, but on the other hand, their concern does reflect some concern from public or from some expert. So we have to be very careful.
And then the next one is, given the diversity and fast evolution of HIV, why not focus on passively administering combinations of broadly neutralizing antibodies? Wouldn't that be more practical?
It is, it is maybe not. Actually, if you think the practicality or the human population need, you don't need to have a passive antibody. You can use the Gilead drug. Now they are very good, can last 6 months. That's almost the same as the passive neutralizing antibody. Okay. But on the other hand, if you look at the detail, do you know how many milligram you have to give human by IV, the monoclonal antibody cocktail? That's very expensive. And also, we are using small amount of people. If you give monoclonal antibody or any antibody at such a high dose, if a long time, you have to give every 6 months time, do you know the potential risk, side effect, and many other things? And the cost itself at this time, after AMP trial, NIH and many other groups developed technically combination of monoclonal antibody. But the issue, as far as I learned, is no company want to do that. It's too expensive.
Agreed. Will you use a new adjuvant for your phase 2A study?
That's a very good question. Uh, Adjuvant, I'm pretty happy with the GRASE. The problem is GRSE is not a licensed vaccine adjuvant. So the business in vaccine adjuvant is you always need to formulate the adjuvant as a part of your vaccine formation move forward. Whether the adjuvant is commercial available as a product, and you have a manufacturer to do that. And that's actually the question facing worldwide vaccine group, not just my own HIV vaccine. So there are people doing CPG, for example, CPG company just bought by a major pharmaceutical company. So the adjuvant, how to use that, why, how to use that, or how to put that into your technology and business development. That's something basic scientists don't think about that, and that's very important. So answer the question to from you toward my HIV vaccine, I worry about that. So I'm also exploring one, who will be my long-term supplier and whether other adjuvant may be readily available. I can use that replace GRSE since my DNA prime and protein boost is so immunogenic. So those are the technical human immunogenicity question, but it is also business questions.
And then is it possible to give plasma DNA as a prime and mRNA as a boost or vice versa?
We did that also. I cannot reveal all the detail. I think you can do some. Whether they can achieve the same thing, I don't know. But we do have a pattern on that.
Yeah, I mean, I guess in principle, it could be similar to using a viral vector together with DNA or RNA, which has been
Yes,
shown to have
yes.
benefits as well. And then here's a long one, so I will read it. I was involved in the development of transchromosomic cattle, in which the bovine Ig genes were inactivated and the entire human Ig gene repertoire was introduced. So they express human IgGs. And I guess the question is, could this model be used to produce anti-HIV human polyclonal antibodies, including broadly neutralizing?
I'm not sure I fully understand the idea. If it's a genetic polyclonal antibody from animal and then but they use a human gene, human antibody, and then like a humanize the animal and the passive antibody to a human, I think technically it's possible. But again, this is following the same question of passive monoclonal antibody, of course, we assume the animal source antibody will be cheap or large quantity, but when you actually deal with and with the this transgenic human, so many other questions. I think working as a scientific thing for next century, that's a wonderful. But in the near future, I guess you will see many, many challenges.
And the last one here is actually a recommendation, not a question. It says you should consider ALFQ as the adjuvant.
Oh, I know this is from Army, right? Yeah, this must be from the Army. We are working with Army. Actually, next week, this week, we have a meeting. They will use my protein and adjuvant. I'm sorry, I'm reveal some secret here. So we'll test that. That's a possible. But again, if I understand correctly, this adjuvant is also not commercially available. So that doesn't solve that business aspect. So I have to, if it's Army, I have to work with Army. Army have to license to some big pharma or some pharma to manufacture. And then they have to do a lot of human study, either with my vaccine or other people vaccine. So that's an example how complicated when you work with a new adjuvant. Yeah.
Good. Well, thank you, everyone. We ran over time, but it was a very good discussion. And thank you again, Sean. Keep up the good work.
Thank you. We will do our best.
Cheers.
Okay, thank you. Take care.